My name is Benjamin. I am joined by Mohammad and Hrithik. In this video, we hope to give you some insight on how randomization occurs in clinical trials. To start, we will introduce the concept of variables.
In an experiment, there are two types of variables, the dependent variable and independent variables. An independent variable is something that the experimenter has control over, and the dependent variable will vary depending on the independent variable.
But wait, not all clinical trials are perfect. Sometimes the dependent variable isn't affected by just the independent variable. Extraneous variables are external variables that impact the relationship between the dependent and independent variable. There are two types of extraneous variables, participant and situational. As an example, consider a clinical trial where researchers study a smartening pill. Test subjects take a pill and are then made to take a test. The independent variable is the dosage of the drug the pill contains, and the dependent variable is the results of the test. Some extraneous variables could be the intelligence of each test subject, their mood, their anxiety, how awake they are, and even the temperature of the room.
The researcher must account for these extraneous variables, and factors that cannot be accounted for are called confounding variables.
As you can see, there are many factors that can influence the results of a clinical trial. Such factors can make it impossible to properly interpret the results of tests. Randomization aims to reduce the effects of those factors as much as possible, as well as permitting the use of probabilistic theory in clinical trials, which forms the basics of statistical tests.
Random assignment of participants in clinical trials helps prevent selection bias. Selection bias is a bias that is introduced when selecting test subjects. Since not all participants are the same, the sample of participants may not be representative of the general public. Randomization also ensures against accidental bias, which are unwanted nuance variables that will affect the results.
In clinical trials, participants are assigned to two different groups, the experimental group and the control group. Groups can be further divided into subgroups for testing purposes. The experimental group is a group of participants that receive the treatment to be tested. The control group is administered a standard treatment, either something already on the market, or a placebo, or even no treatment at all. Random assignment is when participants are randomly assigned to either the control or experimental group. We will discuss simple randomization.
Simple randomization is a single random sequence of assignments. A random generator is used to decide the assignment of a participant. As an example, a coin flip can be used. In this case, heads would indicate a participant is assigned to the experimental group, while tails is the control group. Simple randomization is very easy to implement into experiments. However, if the number of participants is not large enough, randomization results can be skewed, which will result in unequal sized groups. For example, if there are 10 participants and 7 out of the 10 coin flips happen to land on heads. To summarize, randomization is used to thwart unwanted bias and extraneous variables in clinical trials. These biases and extraneous variables can impact the relationship between the dependent and independent variable, which is what a clinical trial studies. We discussed the simplest form of random assignment, known as simple randomization, which is useful for large-scale clinical trials. Thanks for tuning in on this episode of Clinical Trials. We hope to see you again soon.